It's the first time that I am in your beautiful Krakow, and I was impressed by how beautiful this city is, and I would be your ambassador in Greece, definitely. So thank you for your kind words about bone disease, and my expertise and my research interest is the myeloma bone disease. And I'm going to present some of the data. Let's go. Okay, here we are. And of course, you know that multiple myeloma is characterized by the presence of lytic bone disease because due to the increased bone resorption and the impaired bone formation, as you can see here, this leads to pathological fracture, to spinal cord compression, hypercalcemia of malignancy. Before we talk about the pathophysiology of bone disease in multiple myeloma, I want to give you a short background of the biology of bone remodeling. So in our bones, as you know, we have osteoclasts that resorb the old bone and osteoblasts that produce new bone. A very important cell for the bone remodeling is the osteocyte. What is an osteocyte? It's an osteoblast that is entombed into the bone that has produced. And these osteocytes link to each other with this canaliski, as you can see here. Every hour in our vertebra, we have approximately 40 microcracks, as you can see here. When we have a microcrack, then we have the disruption of this canaliski of the osteocytes. This disruption seems to give the first signal, according to the knowledge that we have to date, to the osteoclasts to come to this area and resorb this destroyed bone. At the same time, the osteoclasts give the signal to osteoblasts to come to the area and fill the hole with new bone. But do we know when osteoblasts need to stop? The osteoclasts have given the signal to osteoblasts to fill the hole, but how can they realize that now it's time to stop producing new bone? And this comes from this important molecule, and the name of this molecule is sclerostin. And we know the last couple of years that the osteocytes produce sclerostin in order to make the osteoblasts to stop. And how the osteocytes know that they have to produce sclerostin? Because they feel the pressure of the new bone that the osteoblasts produce. So I'm going to talk only about molecules that new drugs have been created in order not to give you a lot of information about different molecules that may be not of clinical importance. So sclerostin is one of them. And how does it work? This is the different pathways that are important for the osteoblast activation. One is the insulin growth factor, as you can see here, and the other very important is the Wnt type signaling. The Wnt type signaling enhances the production of beta-catenin, which is a transcriptional factor, very important for the proliferation, differentiation, and survival of osteoblasts, and other cells, of course. But let's see what's happening with the Wnt protein. The Wnt protein comes here and binds to this receptor from the Frizzlite protein and the LRP56. So it binds to this co-receptor and it enhances the production of beta-catenin, which is a transcriptional factor. We have two major inhibitors of Wnt. One is sclerostin. Sclerostin binds to LRP56 and inhibits the binding of Wnt to this signaling. So it inhibits the production of beta-catenin and it inhibits the osteoblast function. So sclerostin is an osteoblast inhibitor. Another osteoblast inhibitor is this molecule, DCOP1. It binds to LRP56 and another protein named cremin, and it inhibits the binding of Wnt to the co-receptor of Frizzl and LRP56. So it is a Wnt type inhibitor, and it also an osteoblast inhibitor. So have in your mind DCOP1 and sclerostin, because new molecules targets now, new antibodies, new drugs, targets both DKK1 and sclerostin. And let's go to the bone-forming unit, the osteoclast and the osteoblast. This bone-forming unit is under the control of several systemic factors also. The most well-known is the parathyroid hormone, but also the estrogens, the thyroid hormone. but we have also a lot of local factors, and I'm going to concentrate in two major osteoclast activators, the receptor activator of nuclear factor kappa B ligand, and you need to know this molecule because the new drug targeting rank ligand is denosumab. It is in the market for the osteoporosis management, and also the macrophage colony stimulating factor, as you can see here. Why the macrophage colony stimulating factor is important for the osteoclast? It is important because the osteoclast, as you know, is originated by a macrophage precursor. So the MCSF is important in order to have the increased pool of the macrophage precursors, and then rank ligand, which is produced either by the osteoblasts or by the stromal cells. And we have recently found that the rank ligand in multiple myeloma is produced by the NKT cells. And also we know that T cells and activated T cells produce large amount of rank ligand, and this is why we have osteoclast activity in rheumatoid diseases like rheumatoid arthritis. Rank ligand binds to rank, its receptor, on the surface of osteoclast precursors, and this binding enhances the proliferation and differentiation of osteoclast precursors, but also the binding of rank ligand to rank on the surface of mature osteoclast inhibits the apoptosis of mature osteoclast. Rank ligand is the most important osteoclast function. Without rank ligand, no other osteoclast activator can act, like interleukin-6 or all other molecules. And what's happening when we don't have rank ligand? When we don't have rank ligand, we don't have osteoclast function, we don't have bone destruction, and we have osteosclerosis, as you can see in these rank ligand knockout mice compared with the normal. But as every cytokine in nature, rank ligand has its inhibitor. And this inhibitor, sorry that you don't see the, my headings in the slides, and this inhibitor is named osteoprotegerine. Osteoprotegerine is the inhibitor of rank ligand. It is also produced by the osteoblasts. It binds to rank ligand here, and it inhibits the rank ligand binding to rank. So it inhibits the whole process that I described. So when we don't have osteoprotegerine, we don't have, as you know, anything that can inhibit osteoclast function. The osteoclasts work so much, they destroy the bone, and you can see that in mice that have no osteoprotegerine, we have pathological fracture. You see here this spine with pathological fracture of these mice that have no osteoprotegerine, and this is because osteoprotegerine null mice have low bone volume, this is bone volume, comparing with the osteoprotegerine positive mice. So you can appreciate that the balance between the rank ligand and osteoprotegerine drives the osteoclast activity, and this happens not only in multiple myeloma, as I'm going to show you, but in every bone disease, even in osteoarthritis, where we have published that OPG is increased, or in rheumatoid arthritis, where rank ligand is increased and we have osteoporosis, like also osteoporosis that has to do with inflammatory bowel disease and psoriatic arthritis. So what's happening in multiple myeloma? We have skeletal destruction due to increased osteoclast activity, which is not accompanied by a comparable increase in bone formation, and we have lytic lesions. And let's see how this happened. When myeloma cells binds to the bone marrow stromal cells through addition molecules, we have the production of numerous cytokines that have osteoclast activity. Just to mention two of them, the interleukin-6 that you know that is an osteoclast activator, a very potent one. We knew it from when I was a student in medicine, that interleukin-6 have increased osteoclast activity. But we know that myeloma cell produce these cytokines, the macrophage inflammatory proteins 1-alpha and 1-beta. I told you before that osteoclasts are originated by a macrophage in it. So these macrophage inflammatory proteins are produced by the myeloma cells to attract these macrophage precursors in the microenvironment of multiple myeloma. So, but as I mentioned before, the most important cytokine is rank ligand. In the myeloma microenvironment, rank ligand is overexpressed. Myeloma cells also produce rank ligand. On the other hand, how about osteoprotegerine? Osteoprotegerine is downregulated because of this adherence of myeloma cells to the bone marrow stromal cells. They tell them that you have to produce low OPG, and this lower amount of OPG are further lowered because the myeloma cell binds OPG with this molecule named CD138 or CDKN1. They bind to OPG and they internalize it and degrade OPG. So the ratio of frank ligand to OPG is in favor of frank ligand, and this causes the enhanced activity of osteoclasts that they destroy the bone. Can we measure bone destruction products? Yes, we can, because we know that the organic substance of the bone is the collagen, and specifically collagen type 1, because we have 19 different types of collagen. And these collagen type 1 degradation products, namely amino or carboxytelopeptides of collagen type 1, this carboxytelopeptide is produced by the cathepsin kappa pathway, because osteoclasts can resorb the bone through two pathways, the cathepsin K or the metalloproteinases, and the 1-CTP are produced by the metalloproteinase pathways. And we can measure all these molecules either in the serum or the urine of our patients. Furthermore, this enzyme, the 5-benzo enzyme of tartrate-resistant acid phosphatase, is only produced by activated osteoclasts. So we have increased bone destruction. How about bone formation? The myeloma cells produce DCOP1. I mentioned in my initial biology slide that DCOP1 is a strong Wnt inhibitor and an inhibitor of osteoblasts. And we can see that in multiple myeloma we have reduced levels of bone alkaline phosphatase and osteocalcin, two enzymes that are produced by osteoblasts. But as I mentioned before, and we had an oral presentation at the last AS meeting about sclerostin, and we are going to present in this AS meeting about activin A, the bone marrow stromal cells and mainly the osteocytes produce sclerostin in myeloma that also inhibit the osteoblast function. We don't know if this production of sclerostin is because of the DKK1 production of myeloma cells, because there are also reports saying that the DKK1 makes the osteocytes to produce more sclerostin. But the result is that the osteoblasts are exhausted in multiple myeloma. They don't have a good function. And we also know that the DCOP1 indirectly acts to osteoclasts because it enhances the production of frank ligand, and this was shown by my good friend John Sonessy from Leeds Rock. So the whole thing seems to produce molecules like interleukin-6 that are growth factor for the myeloma cells, because someone can tell you why the myeloma cell spends so much energy to activate osteoclasts. You see all these cytokines and to inhibit the osteoblast function. This is because the myeloma cell has a growth advantage because of that. I want you to remember these molecules, not because I like them, because these molecules have now new drugs that target these molecules. The rank ligand is in the market. It has been licensed even in EMEA, in Europe, for the management of postmenopausal osteoporosis. It has been licensed in the FDA for the management of breast and prostate cancer with bone metastasis, and we expect the results from a big myeloma study with more than 1,400 patients. This is the first. This is the second, DKK1, which inhibits the osteoblast. We have a new molecule targeting DKK1. It's produced by Novartis. The name is BHQ880 and is in phase 2 trials. And anti-sclerostin is produced by Amgen and is in phase 2 studies for prostate cancer. For the management of myeloma bone disease, we have bisphosphonates. As I mentioned, osteoclast is important for the bone destruction, so bisphosphonates inhibit the osteoclast function because they induce the apoptosis of mature osteoclasts, but also they inhibit the differentiation and proliferation of osteoclast precursors. So bisphosphonates today are the cornerstone of the management of myeloma bone disease, and zoledronic acid and pamidronate are the two most in use intravenous bisphosphonates for the management of myeloma. But unfortunately, during the last years, we know that mainly zoledronic acid produced this side effect, which is called osteonecrosis of the jaw. And we see this in approximately 5 to 7% in different retrospective studies. We have 2 prospective studies that shows that this happens in approximately 1% per year. So if we expect that the patient with multiple myeloma lives between 5 and 7 years and is in continuous treatment with bisphosphonates like zoledronic acid, you are going to see that thing. So we expected that possibly with denosumab, the anti-rank ligand, we are not going to see something like that. But also with anti-rank ligand, we have the same incidence like zoledronic acid of osteonecrosis of the jaw. How about proteasome inhibitors? Because my main talk has to do with the proteasome inhibitors and the bone disease of multiple myeloma. This is proteasome. Proteasome is found in all eukaryotic cells from yeast to man, and it degrades proteins. We know that approximately 80% of proteins in the cell are degraded through the proteasome. And let's see one of these proteins, which is the nuclear factor kappa B. Why I'm choosing this molecule? Because the rank ligand is the receptor activator of nuclear factor kappa B ligand. So is the receptor activator for this molecule, for NF-kappa B. So the nuclear factor kappa B is bound to the cell with its inhibitor I-kappa B. In order this group of proteins to go into proteasome, it has to be signed, and this signal gives from these ubiquitins. So this goes into the core of proteasome, and proteasome degrades the I-kappa B, the inhibitor. So the NF-kappa B is released in order to go to the core of the osteoclast, as you can see here. Rank ligand to rank, it enhances different pathways, but also the NF-kappa B, because the receptor activator of NF-kappa B. NF-kappa B is a transcriptional factor that is important for the proliferation and differentiation of osteoclasts. What is going to happen if we inhibit the proteasome? And this is with the green color, this is bortezomib. Bortezomib is a proteasome inhibitor that is used for the management of multiple myeloma patients. Then this NF-kappa B, I-kappa B complex, even if it has been signed with ubiquitins, cannot be into the core of the proteasome because this core has been invaded by bortezomib. So the NF-kappa B is not released, and the I-kappa B is inhibiting NF-kappa B. So what we expect from the osteoclast point of view, we expect that we don't have osteoclast activity if bortezomib is given for patients with multiple myeloma. And indeed, we have several papers published in 2007, 2008 showing in vitro that bortezomib inhibits osteoclastogenesis. And this is very logical because it inhibits the NF-kappa B pathway, which is important for osteoclasts. However, from these publications, mainly from Nicola Giuliani and Batatube Oyazombi from the Pavia University and from MD Anderson, they found that bortezomib also in vitro increased the osteoblast differentiation and the osteoblast numbers. And Gareth et al. Had shown some years before that different proteasome inhibitors increased the bone formation activity. In mice, Penici et al. Showed last year in American Journal of Hematology in mice that myeloma mice who respond to bortezomib managed to increase their bone mineral density of approximately 60%, comparing with non-responsive mice, myeloma mice, who reduced their bone mineral density because myeloma was active. And Mauricio Zangari had shown in the APEX trial, where bortezomib was compared to dexamethasone, that patients who received bortezomib had an increase in the total alkaline phosphatase. Ulrike Heider from the Oran Cesar group of Berlin, now Oran is in Hamburg University, showed that when we treat patients with bortezomib, then you can see that the bone alkaline phosphatase and osteocalcin, if you remember my initial cartoon with biology of bone disease, these are produced by osteoblasts. This production of this enzyme is increased in patients who received, as you can see here, bortezomib, comparing with patients who received conventional treatment with adriamycin, dexamethasone, or melphalan, prednisolone, or even thalidomide-containing regimens. That is why we have done a prospective study with 34 patients with relapsed multiple myeloma. We treated it with bortezomib, as you can see here, and here is the response rate of approximately 65%, and we wanted to check what happened in their bone metabolism. And first of all, we've seen that this degradation product of collagen, the CTX, was increased in patients at baseline compared with controls, meaning that patients with relapsed myeloma have increased bone destruction, and this was dramatically reduced after 4 and 8 cycles of bortezomib therapy. Similarly, the 5B enzyme of TRAP, which seems to be produced by the active osteoclast, was increased here before treatment, comparing with controls, and was also reduced after 4 and 8 cycles of therapy. So we have the human results showing that if we give bortezomib, we have a reduction in osteoclast function, and we have a reduction in the bone destruction. So this confirms the in vitro results about bortezomib and osteoclastogenesis. And this mainly because we've shown that the rank ligand to osteoprotein ratio, which is very important for the osteoclast function, is elevated at baseline comparing with controls and was dramatically reduced after 4 and 8 cycles of bortezomib. Let's go to bone formation. Bone alkaline phosphatase is produced by osteoblasts. Reduced levels at baseline comparing with controls, and you see that then we have increased after 4 and 8 cycles, 50% of increase after 8 cycles of bortezomib therapy. And osteocalcin, which is the other enzyme that is produced by osteoblasts, also reduced at baseline comparing with controls, and you see here 154% of increase after bortezomib therapy. So this gives you the biochemical evidence that we have an increase in osteoblast function. And we've seen that this increase was mainly in responding patients, as you can see here, but also non-responding, but because we had only 9 non-responding, this difference was not statistically significant. But for osteocalcin, you see that also responders and non-responders to bortezomib had an increase in bone formation function. So this means that bortezomib has a unique effect on osteoblasts, and not only an effect on osteoblasts because it kills the myeloma cells that inhibit the osteoblast function. And this was happened mainly because bortezomib reduces the levels of DCOP1 protein. If you remember my initial slides, DKK1 is a Wnt inhibitor, is overproduced by myeloma cells. And you see that here in patients with relapsed refractory myeloma who have increased levels, you see dramatic increased levels of DKK1 compared with controls. And we have also a strong reduction of its levels after 8 cycles of therapy. And you see also then we had an oral presentation at TAS meeting and a poster presentation at European Hematology Association meeting that sclerostin also is reduced after bortezomib treatment in both responders and non-responders when Velcade was given with dexamethasone. Nicola Giuliani showed that in responding patients with bortezomib, you see an increase in osteoblast numbers. So we had the biochemical evidence from my study that we have increased osteoblast production, and this is increased osteoblast production. Nicola showed that it was due to the increased numbers of osteoblasts. You see here with this color that we have increased osteoblast numbers in patients who responded to therapy comparing with non-responders. So bortezomib increases osteoblast numbers, and through this increases osteoblast production and possibly new bone because it reduces the DCOP1 protein. And here is the evidence that these all biochemical things are translated into a beneficial effect for the patients. And we have just presented in the July issue of Annals of Oncology that patients who received bortezomib managed to increase their bone mineral density in their lumbar spine. And indeed, 4 out of 27 patients, meaning 14% of the patients, showed at least 10% of increase in their bone mineral density. And this is very important because if you know about bone mineral density, to increase this within 6 months to have 10% of increase is something very important. And here is the pictures from Mauricio Zangari paper with micro-CT scans where the bone volume in patients who received bortezomib, here you see an example, goes from 12 to 90%. And this was submitted to Hematologica, and I can tell you because I know the result, it has been accepted for publication. It's going to be appeared in one of the next issues of Hematologica. And here are the results of the VISTA study. I don't know if you are familiar with that. Maybe you are not myeloma people. So in this study, there was a comparison between the standard melphalan prednisolone, the standard treatment for patients who are not eligible for a transplantation. And this standard of care for many, many years, since 1960, was compared to melphalan prednisolone again, but with the addition of bortezomib that I mentioned before. So that we see here that the total alkaline phosphatase was, when bortezomib was into treatment, was increased, and of course it was reduced after a while, mainly because of melphalan, which is toxic for the osteoblast, but you see this increase. And sorry for that, but if you see here the DKK1 protein with VMP and MP, I wanted to see that when bortezomib was into the combination, we have a reduction of, as you can see here, minus 694, a great reduction of DKK1 protein, which is an osteoblast inhibitor, but you have an increase only with MP, because also melphalan and prednisolone are toxic for the osteoblasts. And some examples from the VISTA trial that I'm the second author with Michel Deforce that we have submitted that to Hematological a few days before. You see here before the treatment with bortezomib, these vertebra that have been destroyed, and you can see some signals of creation of new bone. And the most, one of my patients, and it's very important, you see here in the skull of this patient, the lytic lesions of myeloma, and after 9 cycles of therapy with MPV, you see that most of these holes have been disappeared. However, as you can see here, when Velcade is given with dexamethasone, yes, we have this beneficial effect in the bone formation that thalidomide dexamethasone cannot produce, but when Velcade is given with thalidomide, here you don't see this effect because thalidomide is very toxic for osteoblasts. And with this VMDT regimen that we published in Leukemia 2 years before, we have 3 toxic drugs for osteoblasts: melphalan, dexamethasone, thalidomide. So bortezomib alone could not manage to override this toxic effect of these 3 drugs. I'm going now to show you a randomized study that has been accepted for publication in leukemia from our group. We have given in this cohort of patients according to peripheral neuropathy status. These are relapsed refractory patients. They received either the combination of lenalidomide plus dexamethasone. Lenalidomide is an immunomodulatory drug that is given for the treatment of relapsed myeloma, or the combination of Velcade plus lenalidomide and dexamethasone. So the results are in this paper, which is in press, but I'm going to talk to you about the bone results. So you see here that when we give lenalidomide plus dexamethasone alone, comparing the change in rank ligand after 6 cycles of therapy, you see that we have almost no change, 0%. But when bortezomib is into the same combination with RD, you have 50% of reduction of rank ligand, and of course, almost 40% of reduction of rank ligand to OPG. This means reduction of bone resorption because reduction of osteoblast activity. How about DKK1, which is the osteoblast inhibitor? You see increase with RD, 40% of increase. And John Sonessy also showed that lenalidomide can enhance the expression of the DKK1 gene. So our results are in accordance with the unpublished data of John Sonessy. But when Velcade is into, you see 70% of reduction of DKK1, meaning Velcade can make the osteoblast to be active in multiple myeloma. And how about bone alkaline phosphatase and osteocalcin, the enzymes that are produced by osteoblasts? With RD, we have lenalidomide dexamethasone, we have 10% of reduction or no alteration at all after 6 cycles of therapy. But when Velcade is into, we have 50%, 100% of increase, meaning that Velcade is able, yes, to enhance the osteoblast function. And this was also an oral presentation at the last ASH meeting. So in conclusion, DKK1, bone disease is a frequent and debilitating complication of multiple myeloma. We know very well that in this pathogenesis, rank ligand, DKOP1 protein, and sclerostin play important role. We have preclinical and clinical data indicate that bortezomib reduces bone resorption, but the most important thing is that bortezomib is the first and only agent to date that increases also bone formation due to reduction of DCOP1, reduction of sclerostin, and possible other osteoblast inhibitors. We have also shown increases on bone mineral density, and we have seen healing of the lytic lesions in subsets of myeloma patients. However, long-term bortezomib studies with clinical endpoints like skeletal-related events and bone mineral density are needed. Before thank you for your attention, I would like to thank all my colleagues in my Department of Clinical Therapeutics, and mainly my chairman, Professor Dimopoulos, and my colleagues in London, Amir Ramtullah and Jane Apperly, in Sheffield, Peter Croucher, and Donna Heath, in Hersted University in the US with Alan Lipton and Kim Legion, in Berlin now, in Hamburg, Oran Cesar, because together with all my colleagues in Athens and Thessaloniki of the Greek myeloma study group, we have produced the results that I have presented to you. Thank you very much for your attention.